In this video, we're going to be introducing the concepts of translation of genes. So we have already talked about translations in the context of the genetic flow of information. We have already established that when it comes to translation in prokaryotes, it happens in the cytoplasm. In eukaryotes, it also happens in the cytoplasm. Remember that in the process of translation, we're going to have as a template the molecule of mRNA. Then the ribosome is going to be what is going to be catalyzing the synthesis of the protein. We're going to have tRNAs bringing the amino acids, and all of those steps that we are going to be at least introducing are going to lead to formation of a polypeptide. So I just want to write a note. Nucleic acids are known as polynucleotides. And proteins are known as polypeptides. I just want to remind you of those terms so there is no confusion of their use in later conversations. So when we talk about the synthesis of proteins, we must first introduce the concept of genetic code. The genetic code is going to be how the nucleotides that are going to be present in the RNA molecule are going to be read. And this reading, remember, that is going to be done by the ribosome. And basically what happens is that we're going to be reading 3 letters. And when I mean letters, these are going to be the nitrogenous bases. At a time. And this 3 letters is what's known as a codon. Now, understand that when it comes to the genetic code, we know it as a redundant code because most amino acids are going to be encoded by multiple codons. The mRNA, and remember that the mRNA is the template for translation, is read from the 5' end to the 3' end, and the protein that is synthesized is going from the N terminus to the C terminus. N stands for amino terminus, and the C terminus is the carboxy terminus. So another word for redundant is degenerate. So what this implies, as I just mentioned previously, is that there are going to be several 3-letter codes that are going to encode for the same amino acid. There are a few amino acids that are exceptions to this, and they are here in these yellow-orange blocks. So, the only two amino acids that are not part of this degenerate or redundant code are going to be tryptophan and methionine. They are the only amino acids that only have one codon defining them during protein synthesis. All the other amino acids out of the 20, so there's 18 amino acids that are going to have different codons in order to define them. I should also establish that there are going to be some codons that do not code for protein, or a code for an amino acid, I should say. So I'm going to highlight them in green, and these are UAA, UAG, and UGA. They are known as stop codons, and they do not encode for an amino acid. As we're going to see once we study the process of translation in detail, these are going to be specific codons that tell the release factors that the completion of the translation has, or that translation has been completed, is what I'm trying to say. So, when it comes to the process of translation, we need to introduce the molecules that are involved in the process of translation, okay? And we already talked about how amino acids need to be incorporated onto tRNAs in order to be brought to protein synthesis site. We have established already that the 3' end of our tRNA molecule is where the amino acid is linked. In addition to that, we're going to have a portion that is called an anticodon. And remember that that anticodon is going to be a sequence that is complementary to the codon in the mRNA. And that's how the tRNA knows which amino acid is going to be incorporated into the growing chain for a protein. So, when it comes to the incorporation of amino acids onto tRNAs, understand that all of this is performed by the enzyme amino acyl tRNA synthetase. So this means that this enzyme is going to be a ligase. Remember, when this process happens, what we have is that this synthetase must match the correct tRNA to the correct amino acid. This process additionally requires ATP. So you can imagine that this is not a trivial process. So as you can see here, we have an amino acid. And this is just a generic amino acid. When we have the tRNA, okay, molecule in the process of ATP, here ATP is being used as an energy source for the reaction. What we have is that now we have the amino acid bonded to the tRNA. As I expressed in previous lectures, the functional group, even though it's not illustrated here, I'm just going to place the oxygen, forms an ester group at the 3' end of the tRNA molecule. Understand that once amino acids are incorporated into tRNA, specifically methionine, when it is incorporated into a tRNA, it's the amino acid that is going to be even further modified. Okay, so Here we have methionine bonded to its tRNA. So understand that methionine is undergoing an N-formal methionine tRNA formation reaction. And as you can see here, what we have is that the enzyme, and by its name you can tell that it is a transferase, is going to be transferring an N-formal group onto the amino side of our methionine. So as you can see, an N-formal group is going to be this group that in essence, as you can see in terms of functional group, is going to make an amide. And then the formal group is what I circled. Okay, so this process happens in E. coli and other prokaryotes. If we rationalize why is it that specifically methionine of all the amino acids have to undergo this further modification once it is incorporated into the tRNA, is because methionine, as we're going to see once we describe all the steps, is the first amino acid incorporated onto protein sequences. That's why it's called the start site. Now, the reason why we need to put that formal group in the amine side of the methionine is because the formal group prevents the amino terminus from reacting and extending protein at that site. You need to be reminded that when protein synthesis happens, and we're going to be exploring it later, the amino group is going to bind a carboxylate group. So adding this formal group to the amino side of methionine, then now in essence you are preventing the amino group from reacting further with other carboxy terminus of amino acids that may be in the cell.